Attached. Okay. So let's get into mitosis. We'll skip that. We'll talk about the role later on. Somebody put up just there that I passed my algebra test is a good way of remembering prophase, metaphase, anaphase, telophase. The I bit is a bit because I do the same thing. I party Monday and Tuesday. It's not my memory aid. Somebody else gave that to me as well, or I don't know where I found it, but it does stick. I party Monday and Tuesday. Here's the thing though, that I is not part of mitosis. So be real careful that the interphase bit comes before mitosis starts. PMAT is the sequence of events when it comes to mitosis. So there's 4 phases. We've got prophase, metaphase, anaphase, telophase. Let's just look at prophase first of all. I'll do it this way around. Now, prophase is a funny one, right? Because the way to think, a lot of what goes on in prophase. So the way you want to think about prophase is that what's happening, what sort of state, or what's the situation with the cell at the very beginning? And then a good way to think about it is, by the end of prophase, what's happened? Okay, so here's what the story for prophase. By the end of prophase, the chromosomes will have condensed. So that's the first point. The nuclear envelope will have melted or disappeared or broken down. And the spindle fibers, these are these structures that are emerging from the ends of the cell, or from 2 different locations in the cell, and growing in towards the center of the cell. They're called spindle fibers. We'll see what they're about in a second. They have started to grow, to lengthen or grow or emerge or whatever you want to talk about. So they're the key things that we want to talk about with prophase. I should say, a really good way of checking your learning when it comes to mitosis is to draw yourself up a kind of a table or a list of prophase, metaphase, anaphase, telophase, and for each of those stages, track the following 3 things. Track what's going on with the chromosomes, what's happened with the nuclear membrane, and what's happened with the spindle fibers. They're the only 3 things you need to be concerned about. The rest, any other detail you know about, forget about it. You don't need it. Forget about it might be it. You don't need it, at least for answering exam questions. Don't, if you know more about mitosis, don't go forgetting it. That's brilliant. So you've got 3 things to track as we go through this. So by the end of prophase, the chromosomes will have condensed from the loose chromatin form that we know them in. The nuclear envelope will have broken down, disappeared, and the spindle fibers will have started to emerge and grow towards the center of the cell. Okay, there's your 3 things. Next stage is metaphase. So metaphase, again, is characterized by, and we think of what's happened by the end of metaphase. By the end of metaphase, the chromosomes have migrated towards the center, what we call the equator of the cell, which is the middle of the cell. The spindle fibers have attached to the chromosomes at the centromeres. The nuclear membrane, well, there is no nuclear membrane at the moment, right? So the nuclear membrane there's nothing we can say about the nuclear membrane for metaphase. Now, one thing I want to point out, if you've noticed, this cell starts off, it's got 3 chromosomes. Just to point that out for you, this is in prophase. It's got 3 chromosomes. Each of those consists of 2 sister chromatids. So we've got 3 chromosomes. Down in the left-hand corner, you'll see that the only way this works is that this is a haploid cell. So the chromosome number is 3, N is 3, and it's haploid. It can only be haploid, right? Because it's an odd number of cells, odd number of chromosomes. It's 3 chromosomes now lined up at the equator. Spindle fibers have attached at the centromeres. That's the end of metaphase. Anaphase, the hallmark of anaphase, is that the spindle fibers contract or retract, they shorten, and as they shorten, they pull apart the sister chromatids. Okay, so now there's one set of what were sister chromatids being pulled towards one end of the cell, towards one pole of the cell, and there's another set of sisters, if you like, individual 3 sisters that are being pulled by the spindle fibers towards the other end of the cell. Here's the really thing that you need to get your head around. At the moment, in this picture, there are 6 chromosomes, right? Because as soon as those sisters are pulled apart, we now refer to them as individual chromosomes. So there are 6 in this picture. In this last picture, there were only 3. And it's not magic. It's just that we've changed now the naming of what we're looking at. At the moment, each of those X-shaped yolks is a chromosome, and it consists of 2 sister chromatids. As soon as they get pulled apart, they now get upgraded. We now refer to them as chromosomes. So now there are 6 chromosomes in this picture. One set of 3 is getting pulled to one end of the cell, and the other set of 3 is getting pulled to the other cell. So now we begin to see what's going on with the the seeming magic of, you know, a cell with 3 chromosomes splitting in 2, and now suddenly you have 2 cells with 3 chromosomes. Well, that's how it happened, because now we're looking at 2 nuclei during what happens in telophase, where the nuclear membrane reforms around each of the sets of the individual sets of chromosomes. The spindle fibers have retracted, shortened, and are shortening, and the chromosomes during telophase, they will start to loosen again, and they will start to return to that loose chromatin, stringy sort of form that we talked about of DNA and protein. But before they do, you'll have seen that they are now fully fledged individual 3 chromosomes in each of the 2 nuclei. So these are still, each of those nuclei is haploid. It has n equals 3. It's got 3 chromosomes, and it started out that this cell, the original mother cell, had again a haploid number of 3, if you remember at the beginning of our story. Now just have a look at this, where now we're going to look to see what happens if you've got a diploid cell. This is a diploid cell. It's got 4 chromosomes, but there are 2 pairs of homologous chromosomes in this. So one pair is brown, and the other pair is kind of, I don't know, beige in this picture, if you can see that. Yeah, somebody's just asking, okay. I wasn't going to mention the word. I'm going to mention it. Did anyone notice those weird green cylinders in the pictures, right? And they're not labeled. And somebody's asked, where do those spindle fibers come from? And that's a totally good, fair question to ask. And they come from those structures, those cylinder structures at the end. Now, we don't need to know about those structures, but some of you, I know your teacher wouldn't have been able to help themselves but tell you that they're yet another C word. So if you've heard the word centrioles, that's what they are. That's where the spindle fibers emerge from. But it's not in our syllabus. We don't need to know about that. And for me, it just adds another word that we actually, it just complicates our life a little bit and maybe causes a bit more confusion. But it's a really good question. So that's where they come from. They come from those structures. So back to our diploid cell. So here's a diploid cell. Look at what happens in prophase. In prophase, we have 4 chromosomes, 2 pairs. 1 pair is in brown, 1 pair is in beige. During prophase, the nuclear membrane this prophase, you notice it's not to the end of prophase yet, because the nuclear membrane hasn't fully disappeared, but it will have fully disappeared by the end of prophase. The spindle fibers have started to emerge. There's another annoying C word, ignore that. The spindle fibers have started to emerge. That's prophase. Metaphase, down on your left-hand side. During metaphase, the spindle fibers will have lengthened and lengthened. The individual chromosomes will have lined up along the equator, and by the end of metaphase, they'll have fully lined up, and the spindle fibers will have attached to the centromeres. Anaphase, top right, the spindle fibers retract. Sister chromatids get pulled apart. Now there are now 2 sets of 4 chromosomes that are migrating, getting pulled to the opposite ends of the cell by the shortening spindle fibers. And then in telophase, new nuclear membranes start to form around each of those sets of 4 chromosomes. And maybe, but not necessarily, during telophase, you get the beginning of this cytokinesis event where the cells start to, there's a cleavage furrow where they start to split in 2. Okay, but not necessarily. So that's what happens when you have mitosis of a diploid cell.